sanctuary is down into the bank. You can tune in live following some links on that education page with Ocean Exploration Trust, or you can see past events through recordings of those Facebook Live or on the YouTube channel. So even if an event happened in the past, you can still take advantage of getting to listen to that at home. You can learn about the meteorites, the dives at Olympic Coast. You can meet some of the team that helps to study different components of the ocean in partnership with Ocean Exploration Trust on the Nautilus, as you can also learn about the Central Coast dives that we have completed and upcoming dives. So I encourage you to check that out. Jen, you were part of one of those events, right?
Yes. It was Chad and I think Sam was the host. And it was just before, it was right after Olympic Coast. And I think they were done with mapping, and I think they were transiting back to Central California and Monterey Sanctuary.
Front row, just a reminder, we're going to do a Niskin at 200. Yeah. Copy that.
Yes, and I'm not quite certain if, because you were breaking up a bit, Jen, but if you go on the nautiluslive.org website and you look under education and then events, look for Explore from Home, and there you'll see all of the upcoming events. Let's see, what is the date today? The 18th. So the 19th, so tomorrow, is the Channel Islands National Marine Sanctuary Santa Lucia Bank interaction. And I believe they have it on Facebook as well as YouTube. You don't need to create an account on Facebook.
Roger. It's kind of drifting over me. That's no good.
And it's a live interaction. It's about 45 minutes long, and you can submit your questions in the chat, and they'll answer them live. So it was really fun to be able to do that and interact directly.
He keeps getting out there.
It's going to stay on 060. Is that good, or you want to take it back a little bit?
No, it's
okay.
I don't want him drifting over me.
Yeah.
So.
Jan and Jennifer, what time is that test that you did for us done? This could be very helpful. Where you heard it clean on the backdoor feed, but it sounded terrible on V-Link.
Let me look in the chat and see. I didn't.
Was it done last night during our watch or this morning?
No, it was this morning.
This morning.
When I look at that. I know I recorded it.
All right, should I start getting the arm in position?
Yep.
Al, according to the science chat, it was 0906 Pacific Daylight Time.
Thank you very much.
So we're going to pull another bottle at 200 or 250?
200.
200.
Right, Steve?
Yep.
We're just
200.
That'll be
we're
bottle
just
5.
now starting to see some inverts in the water column. There's some pyrosomes. Sometimes those pyrosomes, those kind of a long, kind of tubular looking organism, they'll chain up and create like a huge long line, many meters long. Yeah, we're getting more and more pyrosomes.
And you can see them in the Argus cam. Oh, pyrosomes are trying to go into the Niskin. Did you see that?
And we're going for the green tag as number 5 on this one, right?
Yep. Number 5.
Go ahead, Brid.
Okay. No, I mean, I've come around, so it's just we're going to have to hold at some point and let it all get settled out.
We're good on time. It's 10:30. And I last saw we had about 15 minutes to the surface, but we've got time for this Niskin pull and the next at 50 meters.
$200.
Okay,
Okay.
do it. Do like you do. Okay,
There it goes.
coming up
I can
again.
just leave it here because it's only, 150.
What are you doing? Oh, yeah.
Next target is 50.
50 meters?
50.
Yeah.
We wonder if anyone can share with us how the Niskin system is set up so that those close when you want them to.
Would one of you guys like to describe how the Niskins is set up?
So I think I'm going to stop at 100 and just settle out because we're going to lose nav, and I don't want to lose nav when we're doing all these crazy
Roger.
maneuvers.
No, I think the pilots are deep in concentration now.
So basically,
No worries, thanks.
the Niskins basically are spring-loaded. There's a top and a bottom half, or cover, and they're each pulled apart and spring-loaded. And when we pull this little string, basically we pull a pin, and that spring releases, and they snap shut, sealing whatever water is there.
Thank you for that description.
Yeah, a lot of information can come out of those bottles. We can look at the ocean acidification, measuring what is basically the pH of the water, microplastics, nutrients, even DNA of all the organisms that are in the water column at that location. It's a pretty cool tool that we use on large ships, on small boats. Sometimes we can send down just one bottle at a time. Sometimes we have these nice arrays. Sometimes they're attached to a really large CTD, which measures what's basically water temperature and salinity in the water column, as you know, along a depth profile. So they're really cool apparatuses. Not seeing so many pyrosomes any longer. Some people call them
a little
epistles,
patch of them.
because they do look a bit bumpy, especially when you see them on shore. And but they're not really very edible. A little crunchy.
Yeah, I don't know if I know of any humans who have sampled them. I know I have not.
I guess we're
But
okay.
we have.
We're looking good.
Great. Yeah, this is feeling a lot better too. The roll. So Bob, our procedure is also to stop or request the ship to hold position at 10 meters. Is that what you want to do for this as well?
I guess, yeah.
Great. Just want to confirm.
So we're going to close that last Niskin bottle at 50 meters, and then that will be the last sample that we will be taking here in Davidson Seamount area of Monterey Bay National Marine Sanctuary. As Jan was saying, these water samples can be used for many things. We're particularly focused on this cruise.
I was trying to find what eats a pyrosome, but I can't find anything. I would think that maybe mola or sea turtles, but maybe not. No, I can't find anything that actually eats a pyrosome. I do know that when we go out and do oceanographic sampling, the water is almost always a lot warmer than average when we see these very large blooms of pyrosomes. Sometimes you can even see them washing up in the thousands on local beaches, kind of turning the water a bit pink purple.
Yeah, we've seen on the seafloor when they start falling to the seafloor, we see crabs eating them sometimes, but I don't think that's their primary predator or scavenger, I guess.
Can you hear me now?
We can hear you.
Oh, great. I was just going to mention that we did see pyrosomes in the water column out here at Davidson Seamount. We didn't see as many, Jan, that I recall on our descent and ascent over Pioneer Canyon further north in Monterey Bay National Marine Sanctuary. Did you?
Sam, what time do you think we'll be on deck?
We should be on deck in the next 10 minutes.
Ish. We have to stop.
Oh, we have to stop for the
and take
Niskin, yeah.
Yeah, 20.
15, 20?
Yeah.
Came up with Argus cam.
I'm fine if you guys want to just grab it too, and then we can just pull it at 50 and don't have to stop.
We have to stop at 50 anyway to hand over control.
Okay.
Yep.
You can get all set up on it.
Yeah.
Deck van, radio check. Yep, Al's on his way. I need to be here.
I'm going to have to ask again, but do you hear me now?
We can hear you.
Gotcha.
Okay,
Yeah, I do.
great. We are approaching 50 meters where we're going
right,
to
50.
close that last
Yeah.
Niskin.
Pulling it.
Number 6. Maybe I'll also notice that we can see sunlight in the water. Is
Deck,
that-
we're at 50 and we're going to
Oh
take
yeah,
a
it
Niskin.
went.
Roger.
That's all she wrote for sampling.
All right. Thank you, Steve, for doing such a great job helping us manage and keep organized around all the sampling. You've been a great help and resource. Really appreciate it.
Yep. And we'll have we'll be updating the Google Doc spreadsheet with sampling sometime today.
Great.
We're still
Thank you.
entering a little bit of data from the previous dive, but I think it should be done by now, and then we'll post this dive's data within the next probably 4 or 5 hours.
And just
Excellent.
to
You guys
be
are
sure,
awesome.
those the samples we took on this dive will be in the NA123 sampling spreadsheet.
Yeah, these will be in 123. So I did close out the and complete the 122. So if you need to reference any of the material
Does this look
metadata
right
for those samples that we collected on 122, it's
Oh,
all
no, you
current in
gotta
the
bring
Google
your
Drive
shoulder
right
up
now.
more. All the way up.
I think
It's
all.
fine. Leave it. Good.
Okay, up here.
I would say right now for the remainder of the dive, the quad cam is going to give you the best views. They'll be switching back and forth between various deck cameras. And you can really start seeing the light shining, the sun shining down through the water column.
That's right. This is an area where sunlight plays a big role in the food web. There's the base of the food web. There's a bunch of plankton that use that sun, just like plants do on land, to make energy, the phytoplankton, and then they're subsequently eaten by other animals, small animal plankton called zooplankton, which is then eaten by larger things, and on up the food web.
Can I kill Argus lights and thrusters?
Yep.
I do enjoy watching the launches, but I think the recovery is always a lot more exciting because it's just phenomenal how the deck crew it's like a ballet almost. Everybody knows what everybody else is supposed to be doing. They all time it really well. To me, it's fascinating how
Bridge, no?
they just work like clockwork.
Hold
Great
position.
team.
Pilots confirming the MESO has been stopped.
Yeah.
Roger. Roger.
Steve, will you be on the debriefing call?
Lacking in
this afternoon?
clairvoyant
I can be,
skills,
yeah.
you know?
I think we should be, let's see, what time is it now?
I think that's the theme of this dive.
Yeah, I can sneak out. I think we'll be mostly done with sampling, mostly done by 12:30, so I can sneak out there.
Great, thank you.
Roger.
Should I turn off USBL?
Doesn't matter. Might as well have been off for what good it did us.
Ouch. Now we can talk about it.
I know y'all are about to get very busy, and I just wanted to say again, it's really been a pleasure working with all of you on the 8 to 12 watch. Thank you for all your assistance and patience with some of our technological challenges, but all in all, it's been a lot of fun. Thank you.
Thanks, Jen. Thanks, Jan. It's been great working with you again this year.
Yeah, love working with the Nautilus team, no matter which shift it is, but you know, this has been a pretty good shift.
That's right.
All right, Argus is that's a nice
So if I have
stern cam right now.
my druthers in the future, it'll be in person.
Sounds good.
What are we
view
seeing
of
in
the
Argus Cam?
ship. That's the ship propeller.
Wow, look at that.
And we get the other famous view.
There they go. Here's the ballet.
Yeah. And it looks like this is the part where you're saying you're trying to keep the tether taut to help keep Argus more steady for recovery.
You could tell Mark that we might have a wrap.
Deck van, we may have a wrap. Roger.
That was not an approved use of the sea.
He needs the science stick.
Yeah. Roger.
wrap.
Roger, lateral to right. Roger. Roger.
Now's a great time if you're still watching, as Jan said, to put on the quad cam, and you'll be able to see Hercules coming alongside the vessel, and we also see crew on board who are managing that yellow tether that connects Argus to Hercules. And as we heard earlier in our recovery from ROV pilots, how important it is to make sure that that does not get any kinks or bends in it.
There you go. That's better.